Good afternoon, dear viewers. I am Dr. Amit Bhattacharya, Assistant Professor at Department of Zoology, Ramjas College, Delhi University. Today I am here to present a lecture on fatty acid metabolism, mainly what exactly fatty acids are, and then I will be going into the biosynthesis of the fatty acids, the beta oxidation of the fatty acid, how the fatty acids are broken down so that the energy can be provided, and subsequently I will be touching upon some parts of the ketone body formation also, which are one of the most important energy molecules formed in our body. Now, before going into the fatty acid metabolism, we should know something about the fatty acids and the various types of the fatty acids and the important fatty acids. So, all of us are aware of the DNA, RNA, protein and the biochemicals which are present in our body. So, the DNA study of the DNA is many referred as the genomics over there. Now, when the transcription occurs, it forms a RNA which is called as this transcriptomics, the study of the RNA transcripts, and then these RNAs are translated to form the protein molecules which is called as the proteomics. Now, these protein molecules many a times forms the biochemicals which are called as the metabolites, and study of these key metabolites is called as the metabolomics. So, this is some of the interesting features of how exactly the various studies are taking place. Now, if we see a plethora of the organic compounds which is present in our body, we start with the carbohydrates. So, the carbohydrates are majorly the polysaccharides which are made up of the disaccharides, and two monomers of the monosaccharides, they combine to form the disaccharides. So, the carbohydrates have been classified as polysaccharides, disaccharides and the monosaccharide molecules. While in the case of the lipids molecules, there are triglycerides. So, triglyceride, we will see the triglyceride structure. Triglycerides are nothing but there is a glycerol backbone, and to the glycerol backbone there are addition of the fatty acid chain. So, the fatty acid chain can be C16 carbon, C18 carbon or C20 carbon. Now, these lipids are majorly called as the triglycerides, and the triglycerides, as just we told you, that it is made up of fatty acids and the glycerol. Then comes the proteins, which are basically the peptides and the polymers of the amino acid. So, each of the amino acids are attached to one another with the help of the peptide bonds. So, one after another the amino acids are formed and they are combined to one another with the help of the peptide bond, and this polymer of the amino acids is called as the peptides or the protein molecules. The fourth important organic compound which is present in our body is called as the nucleic acid, which is comprised of the DNA as well as the RNA molecules. So, the DNA is a double helix structure which is called as the DNA double helix structure. Now, these DNA double helix structures are made up of nucleotides. The nucleotides contains a base, there is a ribose sugar and there are phosphates which are present over there. So, the base can be ATGC, while in the case of the RNA, which is a single stranded molecule, instead of T there is uracil which is present over there. So, the DNA and the RNA are forming the nucleic acids over there. Now, the fifth important organic compound which is present in our body is the high energy compounds, which is majorly the ATP molecules. Now, these ATP molecules are composed of the nucleotide as well as the phosphate groups. So, in the ATP molecule, which is adenosine triphosphate, there are three phosphate molecules which are attached by the phosphodiester bonds. So, these bonds are the high energy bonds. So, whenever there is a requirement of energy, these phosphates get released from the ATP molecule to form the ADP, and this energy is subsequently utilized by that particular metabolic process. So, this is how the five important organic compounds are playing a major role in the functioning of our living systems. Now, it is important to know what exactly we eat over the meals. So, the meals contains the fruits and the vegetables, then there are high fats, eggs and the meat products which are also there. Then there are milk products which is there, and the carbohydrates are also present in that particular meal. So, a complete meal is having all the important products or the organic compounds into it, the carbohydrate, proteins and the lipids or the fatty acid molecules. So a meal has to be properly balanced so that none of these components are present in a higher quantity, so that they create some kind of problem in the metabolism, and subsequently the person, if taking that particular component in a higher dosages, may suffer from some kind of syndrome or the disease conditions. Now, what are the starch, proteins, fats are broken down into? Now, the starch which is shown over here majorly comes from these potatoes. So, these potatoes contains the high level of starch. So, with the help of the various enzymes which are present in the metabolism or the digestive enzymes which are present, these starch, which are the polysaccharides made up of saccharides, monosaccharides group, they are broken down, and finally the glucose molecule is formed, which is being utilized by the various metabolic processes like the glycolysis and subsequently growing into the Krebs cycle and the other metabolic processes. Now, the proteins which we take are made up of with the help of the amino acids. These amino acids are attached to one another with the help of the peptide bonds. Again, there are certain enzymes which are present in the body which degrade these amino acids to release the free amino acids from these peptide bonds, and these amino acids can further go into the metabolic processes to form the carbon skeleton, and the amino group is released through the urea cycle process. Now, in the case of the lipids or the fats, as we just discussed, it is made up of a glycerol backbone, and there are 3 fatty acid or 2 fatty acid or 1 fatty acid chain which is attached to it. Now, what happens is, with the help of the enzymes, this glycerol backbones is released, as well as the fatty acids are also released, which are subsequently degraded or catabolized in the various metabolic processes, so that the energy can be released for the proper functioning. So, in today's lecture, we will be concentrating upon the fatty acids metabolism, how exactly they are formed, and subsequently how exactly they are catabolized and anabolized. Now, there are various enzymes in the digestion. So, if we see the figure over here, the saliva contains the enzyme amylase, which breaks down the starch into the maltose. Subsequently, as the food goes through the digestive system into the intestine and the stomach, there are certain gastric juices which contains the enzyme pepsin, which further degrades them. Then the pancreatic juices also falls in, which contains carbohydrases, proteases and lipases. So, these lipases, proteases, carbohydrates are very important enzymes which degrades the carbohydrate, proteins and the lipid molecules present in the food which we are ingesting. So, the enzymatic processes plays a very important role in the proper digestion of the food, and subsequently when they are degraded into simpler products, these products are absorbed and then they are metabolized within the cells like the hepatic cells or the muscle cells for the metabolic reactions. Now, if we see the schematic diagram which is shown over here, this shows the various metabolic pathways which are present in the humans. So, we can see it looks something like a microprocessor in which each of the pathways are interconnected with each of one. So, if we see, there are carbohydrate metabolism processes which are also seen over here, like the glycolysis, Krebs cycle, gluconeogenesis and pentose phosphate pathway can be seen. Then we can see the amino acid degradation pathways like the urea cycle, transamination, deamination process, and the lipid metabolism which contains the lipogenesis process. Then we see the glycogen metabolism also, which is shown over here. So, we can see over here there are various pathways which are interconnected to one another with the help of one or the different intermediates present within these pathways. So, the understanding the human metabolic pathways is a very complex mechanism, and till now we haven't been able to decipher most of the pathways and known most of the pathways well. So, some of the pathways are known over here, which we will be discussing in the next upcoming slides. Now, the metabolism majorly contains 2 broad categories of the pathways. One is called as the anabolic metabolism and the other one is called the catabolic metabolism. So, anabolism is basically the small molecules are assembled into the large one and the energy is required. So, we can see over here there are small blocks which can be said as the amino acids which are interconnected to one another with the help of the peptide bonds and the energy is required. While in the case of the catabolism, large molecules are broken down into the smaller ones, and in this breakdown there is release of the energy. So anabolism requires energy while the catabolism releases the energy, which we can see in the diagrammatic representation present over here. Now, before going into the lipids and what exactly the fatty acids are, we should know some of the biochemical importance of these lipids. One of the most important functioning of the lipids is the formation of the cell membrane. So cell membrane, all of us know, are the dynamic structure in which the protein floats in a sea of lipids. So the lipid component of the membrane forms a very important pathway. So they act as a permeability barrier, while the protein component acts as the transport system of pumps and the channels which are present within bifurcating these lipids by layers. So there are so many model systems which have come over here. So I will show you some of the model systems which describes these cell membranes and how exactly these lipids are forming these cell membranes. Now this is a surface of a soap bubble which is shown. So there are bilayers of these can be seen over here by the detergent molecule. So we can see the polar head which are red in color, while the non-polar tails can be seen in green in color. So this is a structured surface of a soap bubble which can be seen over here. Now this is a tabular representation which shows the major components of the plasma membrane in the various organisms. So what are the various components which are present in each of the plasma membrane of the various cells? So we can see the human myelin sheath contains proteins which is about 30%, while the phospholipids is 30% and the sterols is 19%, and the major sterol types is the cholesterol. While in the case of the mouse liver cells, we can see the protein content is 45, while the phospholipid is 27 and the sterols is 25. Again, this sterol type is the cholesterol, which is a C27 carbon containing molecule. Then there are various yeast cells, paramecium cells and the E. coli cells which can be seen, in which we can see the major component is the protein, while a small amount of the phospholipids are present and a very small amount of the sterols are present, majorly the ergosterol, which is called as the prokaryotic kind of the lipid molecules. Now if we see the tabular representation or the diagrammatic representation over here, this shows the lipid composition of the plasma membrane and the various membrane of the rat hepatocyte. So the rat liver cells were taken out and the various organelles, the component present in the organelles were taken out. How exactly the lipid composition is taking place in each of the plasma membranes of each of the components. So if we see over here, the plasma contains the maximum amount of the cholesterol, while the inner mitochondrial membrane contains the lowest amount of the cholesterol. So we can see with the help of the various diagrammatic representation, the cholesterol is shown in orange in color, then there is chidolipin which is shown, minor lipids which are shown, sphingolipids which are shown in red in color, then there is phosphatidylcholine, phosphatidylethionamine which is also shown over here. So we can see the various components which are seen over here, and the chidolipin is present majorly and in the maximum quantity in most of the organelles within the rat hepatocyte, like the Golgi, smooth ER, rough ER, nuclear ER, liposomal ER, outer mitochondrial membrane, inner mitochondrial membrane and the plasma membrane. So these are the various lipid compositions which are present in the plasma membrane of most of the organelle lipid bilayer which is present in the rat hepatocyte. Now one of the most important model system which has been proposed and have been regarded as the standard model system for the membrane structure is called as the fluid mosaic model, which was given by Singer and Nicholson in the early 19th century. Now this lipid bilayer model structure, if we see, there are phospholipid polar heads which are present, which are shown in blue in color. So these are the phospholipid polar heads which are shown. So there are peripheral proteins also which are present over here, as well as there are integral proteins which are present over here. So we can see the integral proteins is bifurcating this lipid bilayer, and they are present on the either side of the lipid bilayer. So they are acting as the channels and the barriers to transport the molecules from one side of the membrane to the other side of the membrane system. Now this lipid bilayer is having the monolayer of the polar heads, and there are non-polar tails which are shown over here. So there are two of these one present over here. That's why we called it as the lipid bilayer, and subsequently we can see some of the oligosaccharide chains which are attached to these glycoproteins over here in the integral protein. So there are presence of the integral proteins also there, then there are presence of the peripheral proteins. We can see some of the sterols are also there, majorly cholesterols are present over there. So all these lipid bilayer, sterols and these peripheral and the integral protein forms a complex model system which is called as the fluid mosaic model system, because all these components are present in a mosaic kind of representation, and it is having a fluidity, means flexibility. That's why it is called as the fluid mosaic model system from membrane systems. Now the phosphatidylate is one of the common intermediate in the synthesis of the phospholipids and the triacyl glycerols. So we just discussed some of the phospholipids which was shown in the membrane bilayer over there. So the first step in the synthesis of both of the phospholipids for the membranes and the triacyl glycerols for the energy storage is the synthesis of the various components. So we can see over here there is a glycerol 3 phosphate which is present over here. So in the first step this is acetylated by acyl CoA to form the intermediate, and which is again acetylated by acyl CoA to form the phosphatidylate. So we can see by the double acetylation processes and addition of the two acyl groups over here, these glycerol 3 phosphate is forming the intermediate which is called as the phosphatidylate. Now subsequently it goes into the formation of the synthesis of the phospholipid and triacyl glycerols. Now what happens next is these phosphatidylate is subsequently goes into the formation of the triacyl glycerol by another round of the acetylation, and this particular process takes place in the endoplasmic membrane, and the liver is the primary site of the triacyl glycerol synthesis. From the liver, the triacyl glycerols are transported to the muscles for energy conversion, or they are stored in the adipose tissues for the proper storages, and when and where required they are released from these adipocytes to transported to that particular part of the body. So the synthesis of the phospholipids and the triacyl glycerols we just saw is taking place through the help of the glycerol when it is acetylated, and subsequently there is an intermediate which is formed which is called as the phosphatidate, and then the triacyl glycerols are formed, and they are majorly formed in the liver cells. Now one of the most important component in our lipid metabolism is the cholesterols, and many of these cholesterols or the sterols we have seen in the fluid mosaic model systems also. So these cholesterols are synthesized from the acetyl coenzyme A by 3-step processes. Now these cholesterols, which are majorly the steroids, they modulate the fluidity of the animal cell membrane systems, and they act as the precursor for the various steroid hormones such as the progesterone, testosterone, ergosterol, cortisone. So the cholesterol plays a very important role in the functioning of the body system. So cholesterols many a times have been referred by the Nobel lecturers or the Nobel laureates is a most highly decorated small molecule in the biology, and there are about large number of Nobel prizes which are awarded, in which most of the studies are being related with the cholesterol mechanisms or the modulation. Now all the 27 carbon atoms of the cholesterols are derived from the acetyl CoA in a 3-step synthesis processes. So we will take up some of the processes, how exactly these 27 carbon atom skeletons are formed of the cholesterol by the 3-step processes. Now if we see the structure over here, this is a C27 kind of structure which is shown over here, and we just discussed it is formed from the acetyl-CoA structure. So the first stage 1 is the synthesis of the isopentane pyrophosphate, which is a activated isoprene unit that is the key building block of the cholesterol. The stage 2 is basically the condensation of 6 molecules of these isopentane pyrophosphate to form the sequeline, and subsequently this sequeline cyclizes by astonishing reaction, and the tetracyclic product is subsequently converted into the cholesterol molecule. So we can see from the three structures structure how exactly these isoprenes are forming into a sequeline, and subsequently they are getting cyclized to form the tetracyclic molecules, and these molecules are further getting formed into the cholesterol structures. Now, depending upon the precise conditions and the nature of the lipids, there are three types of the lipid aggregates that can be formed when the amphipathic lipids are mixed with the water. So, the amphipathic lipid means it is acting both as the polar as well as the non-polar also. So, that is why the name has come up over here. So, depending upon the precise conditions and the nature of the lipids, and in the presence of when the amphipathic lipids are mixed with the water, there are three lipid aggregates which are formed over here. The first one, if we see, is called as the micelle. So, we can see the hydrophobic chain of the fatty acids are sequestered at the core of these spheres. So, we can see the polar heads and the non-polar tails which are present over here. The second one is called as the bilayer. So, this is an open bilayer which is seen over here. So, we can see all the acyl side chains except that of the edges are present over here. So, we can see the bilayer. So, there are polar heads which are shown blue in color, and there are non-polar tails which are shown over here. So, there are two lipid layers which are formed over here, which is called as the bilayers, and when these bilayer structures are folded, they form a circular structure which is called as the liposomes. So, a liposomes can be seen over here, in which this lipid bilayer circularizes, and in the center there is an aqueous cavity which is present, which is filled by the water molecule. So, we can see over here the liposome structure also, the bilayer structure and the micelle structure. So, they all depend upon the precise conditions and the nature of the lipids and how exactly they are formed in the presence of the water molecules. Now, we go into the fatty acid metabolism, what exactly the fatty acid metabolism is, and how exactly these fatty acids are degraded or formed within our body system. Now, most of the fatty acids, if we see, are taken from the diet sources. So, what is formed over here is there is a process called as the esterification, by which the excess of these fatty acids, they are converted into triacyl glycerols or the fats over there, and they are stored over here. Now, as soon as we stop taking the diet, or there is some kind of abnormal conditions which is present in our body, these triacyl glycerols, they start degrading into the body system, and this particular process is called as the lipolysis. Now, these fatty acids can further be formed from the acetyl-CoA also, which is called as the lipogenesis, and these fatty acids can be broken down by the process called as the acetyl-CoA when the energy is required by the body. Now, these acetyl-CoA can be further formed from the carbohydrate as well as the amino acids also. Now, we just saw that these acetyl-CoA plays a very important role in the formation of the 27 carbon containing molecule, which is called as the cholesterol, and the process is called as the cholesterol genesis. Now, these cholesterol can further go into the formation of the steroids, while the acetyl-CoA can go into the citric acid cycle for subsequent and complete oxidation, or it can go into the formation of the ketone bodies. So, we will be discussing about the beta oxidation and how exactly these fatty acids are formed, and then we will be taking up the ketone body formation also. Now the lipids, the examples which are shown over here for the lipids, the fats and the oils. So these are basically the glycerin or the glycerol plus 3 fatty acids, and they are the energy storage in the animals, while the next one is called as the phospholipids, which contain glycerin plus 2 fatty acids, 1 polar phosphate containing group which is present over here. So they form, these phospholipids are the important component of the plasma membrane or the cell membrane. Then there are waxes, which are the complex hydrocarbon, not a polymer, so they act as the waterproofing for the cell surfaces, especially in the plants. Then there are sterols and the steroids which are formed, so they are not polymers, having distinctive 4-chain 4-ring structures which are present, so they are important for the formation and the for biosynthesis of the hormones, which plays a very important role in the various metabolic processes and the proper functioning of the body sources. Now the fats are the major components which are stored in our body and which is used as the energy and protect us from the cold. So some of the fats which are shown over here, and we will be discussing about the triacylglycerol, which are triglycerides. So we can see there is a glycerol backbone, and to this glycerol backbone there are 3 fatty acids which are attached to this. Now the fatty acids are majorly distinguished into saturated as well as the unsaturated one. So the saturated fatty acid are the one which doesn't contain any double bonds. So there are single bonds which are attached over here. So the various common fatty acids which are shown over here, the 12 carbon containing fatty acid is called as the lauric acid, which has got a melting temperature of 45. Then there is 14 carbon containing fatty acid, which is myristic acid, melting point is 55. Then there is 16 carbon fatty acid, which is called as the palmitic acid, which has got a melting point of 63. Then there is 18 carbon containing fatty acids, which is called as a stearic acid, and the 20 carbon containing fatty acid, which is called as the arabic acid, having a melting temperature of 76 degrees Celsius. Now in the fatty acid metabolism, the other classification is the unsaturated fatty acids. So these unsaturated fatty acids are the one which contains double bonds. So we can see over here the various double bonds which are present in the fatty acid. So there are palmitoleic acid, then there is oleic acid, linolenic acid, arachidonic acid. So depending upon the larger chain and the number of double bonds, we can see the melting point is decreasing. So initially it was 0 degrees Celsius, it came to 13 degrees Celsius, minus 5 degrees Celsius, 11 degrees Celsius, and minus 49 degrees Celsius. So these are the unsaturated fatty acids. Now in the biosynthesis of the fatty acids, these biosynthesis contains the fatty acid synthase multi-enzyme complexes. So these multi-enzyme complexes contains 2 major components. So one is the unit 1, the other one is the unit 2. Now over here, if we see the unit 1 and the unit 2 are having about 7 major enzymes which are present over here, as well as there is a component which is called as the ACP or acyl carrier protein. Along with the over here there is cysteine and there is phosphopantothenic acid which are having the thiol groups over here. Now the unit 1 and the unit 2 plays a very important role to form the fatty acid synthase multi-enzyme complex, and the fatty acids are synthesized one after another in these multi-enzyme complexes. So what happens is, to the unit 1 there is a addition of a acetyl CoA, and to the unit 2 there is a addition of the malonyl CoA, and subsequently they are attached to one another, and the C16 palmitic acid is formed with the help of these multi-enzyme complex or the fatty acid synthase molecule. So we can see over here there are 7 enzyme molecules which are present over here, as well as there is a ACP. So the 7 enzymes are both present in the unit 1 as well as in the unit 2 over here. Now the fatty acid catabolism, which is many a times referred as the beta oxidation of the fatty acids. Now over here I have shown the beta oxidation of the palmitic acid, which is a C16 containing fatty acid over here. Now it is called as beta oxidation because the bond between the alpha and the beta carbon is broken down in each of the step of the fatty acid catabolism process. Now what happens is this particular catabolism process takes place for about 7 successive cycles, and at the end there is release of 8 acetyl CoA which can go into the Krebs cycle or the citric acid cycle. So we can see over here, this is a pyramid structure of the palmitic acid which is shown over here. So in the first cycle what happens is the alpha and the beta carbon bond are broken up, so there is a release of acetyl CoA and the chain get reduced by 2 carbons. So it is many times referred as N-2. So initially we started with 16, now it has become 14 carbon, and subsequently it again goes for another round of the beta oxidation, and this is how the process takes place. Now this is a cyclic representation of how exactly the palmitoyl CoA goes for 7 particular cycle. So in each of these cycle there is a release of a 2 carbon molecule which is the acetyl CoA, and the chain get reduced by 2 carbon molecules. Now at the end we are having 8 acetyl CoA which are present over here. Now if we see the detailed structure of the beta oxidation molecule over here, we start with the palmitic acid. So in the first stage what happens is there is a reduction process which is taking place. There is a reduction process, oxidation process which is taking place over here. So what is happening is the FeD is getting converted to FeDH2, and in this process there is a formation of a double bond which is called as the trans delta 2 anyl CoA. Now subsequently there is a hydration process which is taking place, and in this hydration process what is happening is there is a formation of hydroxyacyl CoA which is formed over here by the hydration process. So we can see the third carbon is getting a hydroxyl group present to it. Now in the next step what happens is again there is a reduction process, oxidation reduction process which is taking place. So the protons from these hydroxyl group are released and they are taken up for the reduction of the NAD to form the NADH plus H plus, and there is a formation of a keto group. So it is called as the beta ketoacyl CoA complexes, and finally what happens is there is a addition of a coenzyme ASH. So the acetyl CoA is released by this particular enzyme which is called as a thiolase, and the chain gets reduced by N-2. So what we are seeing over here is there is a multi-step processes which is taking place over here, and each of the processes what is happening is the chain is getting reduced by N-2, and this particular cycle takes place for 7 times. So what happens is in the 7 reactions we get to release about 8 acetyl CoA, and there is release of formation of 7 FeDH2 and 7 NADH plus H plus ions are formed, and these high energy electron carrier molecules which are formed from the beta oxidation then go into the electron transport chain for the formation of the ATP molecule. Now the beers carry out the beta oxidation in the sleep. So this is one of the most characteristic example how exactly these beer go into the non-hibernating and the hibernating levels. So the beer maintains a body temperature between the 32 to the 35 degrees Celsius, which is the normal non-hibernating levels. So this is majorly maintained with the help of the beta oxidation processes which takes place within the beer. So although they expending about 25,000 kilojoules per day, the beer does not eat, drink or urinate or defecates for months at time. So this is majorly taking place with the help of the beta oxidation, which is helping the beer to maintain the body temperature. So the hibernating bears use the body fat as their sole fuel, and the fatty acid beta oxidation yields sufficient energy for the maintenance of the temperature and the various energy requiring step processes which takes place within the body of these beer. So we can see over here the fatty acid beta oxidation plays a very important role in most of the organism. Now, in the fatty acid beta oxidation, we come into the next topic, which is called as the ketogenesis. Now, the ketogenesis majorly occurs when there is a higher rate of fatty acid oxidation which takes place in the liver or the hepatic cells. Now, what happens is, under certain metabolic conditions such as the fasting or the diabetic condition, which are associated with high rate of fatty acid oxidation, the liver produces three major components which are collectively called as the ketone bodies. Now, these are called as the acetoacetate, beta hydroxybutyrate, and the acetone molecule, which is continuously undergoing decarboxylation and formed from the acetoacetate. Now, if we see the structure over here, and these three components are called as the ketone bodies. Now, the ketone bodies are formed from the acetoacetate. So, the acetoacetate spontaneously decarboxylates to form the acetone molecule, and then the acetoacetate releases its gets reduced to form the beta hydroxybutyrate with the help of the NADH, and in the presence of the enzymes which is called as the 3-hydroxybutyrate dehydrogenase. So, these three major components are collectively called as the ketone bodies. Now, if we see the diagrammatic representation over here, these ketone bodies serves as the major fuel for the extrahepatic tissues. So, when the person is in the starving or the untreated diabetic conditions, these often leads to the overproduction of the ketone bodies. So, there are medical problems which are related, and these ketone bodies are the one which are generally diagnosed in the blood samples, and subsequently the patient goes into the diagnosis and the proper treatment regimes. Now, what happens is, we have just discussed the fatty acid in the liver cells goes into the beta oxidation to form the acetyl CoA. So, in the normal process, what happens is, the acetyl CoA goes into the Krebs cycle for the complete oxidation process. But in the starving and the untreated diabetic conditions, what happens is, there is overproduction of the ketone bodies and there is excessive fatty acid degradation which is taking place. So, the ketone body formations is taking place, and these are taking place with the help of the enzymes, and these ketone bodies are then subsequently, with the help of the blood, they are transported to the various extrahepatic tissues, which serves as the fuel for the metabolic processes in those extrahepatic tissues. Now, this is a diagrammatic representation how exactly these ketone bodies are formed and how these ketone bodies are moved through the blood circulation into the extrahepatic tissues. So, we discussed that from the acyl CoA, the acetyl CoA are formed, which is a 2-carbon molecule which can be formed from the glucose also. In a normal case, this acetyl CoA goes into the citric acid cycle in the liver cell, but due to some metabolic conditions, what happens is, the ketone bodies are formed. These ketone bodies are taken up from the liver cells or the hepatic cell by the blood vessels, and then they move into the extrahepatic tissue. Many a time these ketone bodies, when they are present in a higher quantity, they start coming up in the urines, or they go into the lungs, majorly in the form of the acetones also. So, the urine conditions of the ketone bodies can be also detected, as well as in the lungs the acetone levels can also be detected, where there is a high production of the ketone bodies taking place. Now, the ketone body subsequently goes into the formation of the acetyl CoA, and these acetyl CoA goes into the either citric acid cycle, or they can be formed into some other form. So, these things are majorly formed into the extrahepatic tissues in the body surfaces. Now, when there is an overproduction of these ketone bodies, there are certain terms which are generally in the terms which are being used. One of the term is called as a ketonemia, which means that when the ketone bodies are present in an increased quantity in the bloods, it is called as a ketonemia. Then there is ketonuria, which is the increased amount of the ketone bodies in the urine. Then the third term is called as a ketosis, when there is an increased amount of the ketone bodies in the tissue, especially in the lungs. We just saw that when the smell of the acetone comes in the breath, which means that there is a higher quantity of the ketone bodies which is getting accumulated in the lungs also. Then it is called as the ketoacidosis, which is majorly caused due to starvation or the uncontrolled diabetic mellitus, and the major treatment for to stop these overproduction of the ketone bodies or control them is the administration of the insulin or the diabetic mellitus medicines to that particular patient, and correct the metabolic abnormalities and the associated with water and the electrolyte imbalance also many a times. So, depending upon the levels of the electrolytes and the water's quantity in the body, the doctor recommends that particular dosages also. Now, there are certain reagent strips also which are available in the market, which are easy to use, and they help in the detection of the ketone bodies present in the urine samples. So, these reagent strips provide a very fast, convenient way to test the urines for the present and the concentration of the acetoacetic acid, majorly which is collectively called as the ketone bodies. So, when the substance start coming up in the urine, that means that there is a misfunctioning of the carbohydrate and the metabolism, fatty acid metabolism, which is taking place. And by checking up the coloration with the standard color plates which are present in that particular strip samples, we can say that what exactly is the level of the ketone bodies present in that particular person's urine, and is the person going into a diabetic condition or is a normal conditions which is present over here. Now, the cholesterol also plays a very important role in our body system also. So, we will take up the few of the importance of the cholesterol system. So, we just discussed that the cholesterols are formed from the acetate, the C2 carbon to form the isoprene, then there is sequelin, lanosterols which are formed, and finally the cholesterol is formed, which is a C27 kind of molecule. Now, from the cholesterol there are formation of the bile salts, cell membrane, it plays a very important role in the fluidity of the cell membrane, then the vitamin D are formed, cortisones are formed, aldosterols are formed, testosterone, estradiol. So, various hormones are formed from these cholesterols only. So, the cholesterol plays a very important role in the functioning of the body mechanism. Now, the cholesterol levels have been graded into various categories like the desirable, borderline high and the high levels. So, these categories are majorly defined by the National Cholesterol Education Program cholesterol guidelines. So, the total cholesterol levels which are shown over here, they are mg per deciliter of the blood. So, the desirable values are shown over here, then the borderline high and the high levels are shown over here. So, the total cholesterol level, low density cholesterol, low density lipid cholesterols, which are many a times referred as the bad cholesterol, and the high density cholesterol, which are good cholesterols, are also shown. Apart from that, the triglycerides are also shown over here. So, any value above 400 mg per deciliter is a high level of triglycerides, and the person may be given certain kind of medicines to control these particular levels of triglycerides, because over a period of time accumulation of these triglycerides may cause some kind of problematic, or the total cholesterols may also cause some kind of problem. So, one of the problem which is shown over here is called as the atherosclerosis, which is a major heart problem which is formed by the fatty acid substances, which forms a deposit of plaque in the inner lining of the arterial wall. So, the inner lining of the arterial wall is being plagued by the various these high level of the cholesterol and the triglyceride. Now, what happens is, if we see, this is a cross section of the artery which is normal in condition. So, we can see the artery wall which is normal in condition, and from these artery there is a blood flow which is taking place. Now, in the next picture we can see there is a plaque which is formed over here, and this plaque is the one in which there is a formation of, or the accumulation of the cholesterol or the triglycerides which are taking place. So, what has happened is the lumen of that particular arterial capillary has decreased. Now, by decreasing the arterial capillary, what is happening is the blood flow level increases, the person suffers from high BP condition or blood pressure condition, the pumping rate of the heart increases, and subsequently there are chances that the person may suffer from the heart attack conditions also. So, these are the various conditions which can be seen by the high level of the fatty acid accumulation in our body, especially the triacylglycerides as well as the cholesterols in our body. So in this lecture today we discussed about what exactly these fatty acids are and how these fatty acids are forming the lipids, how exactly these lipids are playing a major role in the lipid bilayers, the lipids layer formations, the cholesterol which are forming the steroids as well as the hormones, and subsequently we discussed and touched upon some of the parts of the fatty acid metabolism, which is the biosynthesis of the fatty acid. Then we discussed about the beta oxidation of the fatty acid and the ketone body formation, and then we discussed about the various blood problems which occurs due to high accumulation. So with this note I come to the end of my lecture. Thank you very much.